Namaste and warm welcome to the YouTube channel RPH BISHNU sir. Today I am going to discuss new topics, that is research and development. How the research and development is come to the new era. Today we are going to discuss with these new topics. Milestones in the development. First of all, if the compound is discovered, then a patent application made and the publication in the scientific journal. Here are the four phases, that is the research and discovery, early development, full development, then pre-market activity. In the early development, there will be the trials in a patients. Then in the pre-market stage, submission of registration dossier to the regulatory authority. Then it will go finally for the registration. How much years that it will take to the time means in the research and discover, it is here it means 0 to 5 years times it will take. Then early development 5 to 8 years, in full development 8 to 12 years, in pre-market activity 15 years times it will take. In the clinical studies, simply we should know the clinical study means the trial should be done in the human beings. Preparation of the initial clinical plan, selection of the clinical study location. First of all, we should do this one. Human trials with the healthy volunteers to the test tolerability. In the clinical study, we should know the safety and efficacy. Large scale trials to determine definitive safety and efficacy in the patients. Same things here. This clinical study also we have divided into the four parts, that is research and discovery, early development, full development, then the pre-market activity. Then we should know the toxicology of the drugs. The toxicology should always carry out in the animals. Clinical trial means the always carry out in the human, and toxicology always carry out in the animals. In the animal, determine the effects of the medicine in animals when administered over 2 to 13 weeks. Here, we should see the mutagenic potential. Determine the effect on the impregnations and the implantation in animals and whether the active substances can affect the fetus. Determine the reproductive effect upon future generation in animals. So, if we go for the research and development means that the toxicology should carry it out in the animals. Longer term animal study, does active substances have the long term side effects? We should observe this type of the effects in the toxicology. In the pharmacokinetics and metabolism, simply the pharmacokinetic we should see here the drug, the molecules, the absorption, distribution, metabolism, and the excreted in the animals, and determine the how and the what extent the medicine is absorbed by the humans. We should see the pharmacokinetics and the metabolism property of in the population such as elderly, different races and sexes also we should see. Determination of the how the medicine is distributed, metabolized and excreted by the human. Final construction of the pharmacokinetic profile of the medicine we should see here in the pharmacokinetics and the metabolism property of the drug while the developments of the new molecules of the drug. Now we can go for the make the dosage form. In the dosage form, we should see the pre-formulation activities or the physiochemical properties also we can know. That examples are the particle size, solubility, etc. And the development of the clinical trial formulation for dosage form for the voluntary trials. Then the process development for the large scale production of the dosage form, testing of the stability of the dosage and the determination of the shelf life. And develop the market formulation based on the known characteristics of the medicine and patient group involved. Example, age and condition, for example, tablet, capsule, injectable or transdermal patches. Then we should go for the process validation and the production of the dosage form for launch. Another thing is active ingredients. The therapeutically active components in a medicine final formulation that is responsible for its physiological or pharmacological actions. Just we should see here batch zero synthesis, the first non-GMP batch, Then batch 1 synthesis GMP standard, batch 2 GMP, batch 3 GMP, batch 4 using final method of the synthesis, final production concept, batch 5 compound produced in the full scale production, equipment and the validation and routine of the production. We should see here like this. Then the marketing phase. In the marketing phase, early stage commercial assessment of the medicine taking into the account medical needs and existing therapy on the market. Marketing input to the design of clinical trials, especially in the choice of comparator medicine. Full development commercial assessment. We should see a selection of the trade name begins. Trade name chosen for the medicine. Profiling development of the comparative studies on, for example, specific patient groups and the other similar medicine, study possible application of the medicine to the other diseases. Then, medicine information formulated into the brochures and the video displayed at the congress and the symposia. Like this, we can do the marketing page. In the pharmacoeconomics page, the cost is the main determined to the value of the dosage form. Here, first of all, we see the research in the that link to the clinical trial phase 2. This is the safety and efficacy, definition of the healthcare cost caused by the diseases, assessment of the potential impact on the new medicine of healthcare cost. And economic evaluation means example, cost saving and cost effectiveness. Compilation and the publication of the pharmacoeconomics results, we should see and decide the cost of the drugs. And how did the regulatory affairs work? Means application for the trial authorization for to begin the trials on healthy volunteer. The authority should be given by the regulatory authority. Then application for the trial authorization to begin trials in the patients, that is the early development. Application for trial authorization to begin trials in the patient full development. Here we see here three things means interaction and with health and the authority should be the authorized by the regulatory affairs. Compilation for the registration dossier, formulation of the medicine labeling and doctor or patient medicine information, interaction with health authority, review of registration documented by regulatory authority. By the permission of the regulatory authority, then only we can market the drugs. If any dosage form, first of all, we should go for the research and development means we should go for the design of experiment. The design of experiment is nothing that is the systemic procedure carried out the under controlled condition in order to discover an unknown effect to test or establish the hypothesis or illustrate unknown effect. Design of experiment has proven to be an effective tool for the formulation scientists throughout the many stages of the formulation process. Here, the O is the optimize, that means making as perfect, effective, or the functional as possible. Like this, we can have to optimize the dosage form technique in the relatively new to the practice of pharmacy when using the intelligently. These statistical methods will broaden the perspective of the formulation process so that we can conduct by the design of experiment. In the design of experiment also there are the advantage and disadvantage. In advantage, a high level of the control and clear cut conclusions and many variation can be utilized. Okay, and inside this, there are the some of the disadvantage also. Large subject to the human error can create the artificial situations, can take an extensive amount of the time to do full research. These are the some disadvantages of the design of the experiment. Ideal characteristics of the drug delivery. There should be the some ideal characters. The control the drug release, that is the novel drug delivery system. Increase the absorption of an insoluble API. Reduce the API's toxicity. Improve the drug release profile. Create the truly best in the class product. Reduce price. The ability to the target. Terms. What are the terms used in the design of experiment? That is the variable. Variable means that is the dependent and independent variables. The independent variables, just like an example, lubricants, concentration of polymer influenced by the independent variables are example, hardness and the dissolution rate are the variables. And the factors. Factors may be the here qualitative and the quantitative. The polymer ratio are here the calculated and the concentrate temperature, lubricants are the factors to affect the dosage form. Just example, a quantitative factor has a numerical value to its, for example, concentration 1%, 2%, or so on. Drug to polymer ratio 1:1, 1:2, etc. That should be the consider the factor. Qualitative factors are the factors which are not numerical value. For example, the polymer grade, humidity, condition, type of equipment are the qualitative factors. These qualitative factors are the discrete in the nature. And the levels. If any design we have the making means there will be the levels. That for example, in concentration factor 1% will be the one level, while 2% will be the another level. And the two different plasticizers are the level for the grade factor. Usually levels are indicated as the low, middle, or the high. Normally, +1 and -1 are the levels. The general formula for the conversion is level x is equal to average of the two levels divided by half the difference of the level, where x is the numerical value. And the response. And the response means the what happened to the dosage form. It means the such example, disintegration time, duration of bioenergy. It is shared for the response to see the dosage form. Effect. The effect of the factor is the change in the response caused by the varying the levels of the factor. This describes the relationship between the various factors and the levels. And the interaction we should see here. Interaction is the similar to effect, which gives an overall effect of the two or more variables factors on a response. For example, the combined effect of the lubricant and the mean factors and the guidance factors on the hardness of the tablet. In the trial and the error method, a lot of formulation have to be prepared to get a conclusion, which involves the lots of money, time, and energy. This can minimize by the optimization technique. And here the design of the experiments, one of the method is the response surface methodology. The response surface methodology and experiment strategy that was developed in the 1950. The RSM has a different method, response surface methodology RSM. These methods are the one example is the central composite design. What happened in the central composite presented by the box emulation is the based on the factorial design with additional points to estimate the curvature of that design. CCD means central composite design encompasses a full factorial or fractional factorial approach which can be represented as shown in below at the 8 corner of a cube. There are the 6 points known as the axial or star points located in the center of each face of the cube, with the final point located in the middle of the cube that is known as the center point. Like this, you have the equal chances of the there to make the dosage form. The factors under the considerations are usually investigate the 5 different levels. They are the minus alpha, -1, 0, +1, and plus alpha. This is the central composite design. Here is the figure from which side we are assuming the formulation means this all those things considered for the clear and the valuable dosage form design should be compared. Another example is the box Benken design. Also there to optimize the formula. The box Benken design describes the class of the second order designs based on their 3 levels. That is the -1, 0, and +1. In this design approach, the treatment combination are located at the midpoint. Yes, the each of the process is placed at the center as represented in the figure below. This is the figure shown in the box Benken design. How to calculate the box Benken design? N is equal to 2k and k-1 plus constant of the replicate number, the center point replicates. Here where n is equal to the number of experiment, k is equal to the factor number, c not is equal to the replicate number of the central point. In order to the design the dosage form is the dollar design also there. In the dollar design also there is the determined by using the equation how this will be the conduct the experiment. There are the different software for design of experiment. The examples are Design Expert, Chip, Multisimplex, Nemorda. Software for general statistical nature are such Minitab, and the newly available in the market is the Sigma technology. And today we are going to discuss regarding the Sigma technology also. These are the valuable persons who are using the Sigma technology in this thesis and the other project. They are the uh, valuable persons using the Sigma technologies. Now we are going to discuss regarding the Sigma technologies. How the Sigma technology is showing the all parts and the features of the dosage form. Simply we are going to discuss in the graph. See, if you keep the numerical value in the software and how to go after the you have optimized the formula, then it shows the ANOVA of the drug release. In ANOVA of the drug release, the how does the dosage form showing the good formulations we can identify the easily. That is standard deviation we can see here. Curvature effect 95% confidence level limit curvature effect also we can see here. How much the data it will show if the value of the hair came means higher the formulation will not work by the end of the drug release we can see here the formulation is working nicely like this the contour plot of the drug release the contour plot also showing by the sigma technology and here the hardness by the ANOVA how which formulation will be the best the we should see here hardness also of these formulations. Here the T50 contour plot means drug half release contour plot also we can see here by the technology by using the software. Thank you very much. Please like, share, subscribe and comment on video. The new video upcoming soon. Thank you very much.